Welcome to our lab podcast on the respiratory system. As we begin, we're looking at a cross-section through essentially the throat, where we can see a cross-section of the esophagus that's present on the right. And again, remember, our esophagus is going to show this highly folded mucosa, our submucosa. These are some blood vessels present. And then when we get into the adventitia, it's going to blend with the adventitia of the trachea. Now, even at this low magnification, we can pretty much guess that we're looking at cartilaginous rings. We have our mucosal lining. We're going to have our glandular material here in the submucosa. And then when we look at this area between the arms of the cartilage, this is going to be the area where we see our trachealis muscle. And so we're going to focus on this area. So looking at the inset, we can see some smooth muscle that is moving into this area. We have many of our tracheal and bronchial glands that are going to be secreting materials. We can see some of the ducts of those glands right here. And then we've got our respiratory mucosa that we see lining the lumen of the trachea itself. And here we can see bits of that cartilaginous ring, the tip there as well as there. Once again, we're looking at the epithelial lining at the top. We can see the seromucous glands in the trachea, the ducts radiating through the submucous. Looking at a different section, different stain as we get into our trachea, we can hear, we don't see the cartilaginous rings, but we're looking at the mucosal lining, the submucosa, these glands and their ducts. This a little bit more easily, we can see the pseudostratified nature of the epithelium. There we can see the cilia present on the apical surface and the goblet cells that have the mucinogen granules staining dark color. Now, so far we have been looking at the extrapulmonary structures. Now we are looking at the intrapulmonary. We saw this in lecture as we look at our bronchus versus bronchiole. The bronchus, remember, is going to have the cartilaginous plates. And we can see that in this case, this bronchus has cartilaginous plates completely surrounding it. And at this histological level, we're simply going to call this an intrapulmonary bronchus since we are within the lung tissue itself. Here we have a bronchus. Here we have one, we can still see the cartilage. So this is also going to be a smaller, but still an intrapulmonary bronchus. Higher magnification, we can begin to see the cartilaginous nature of the histology of our cartilaginous rings. We can see the slightly folded nature. You really at this magnification can't make out our respiratory epithelium, but if you could get close enough, that's what we would see. And we begin to see our spiraling smooth muscle, and we still have our bronchiolar glands that are present in the submucosa. Again, taking a closer look, here we can see even more so our typical hyaline cartilaginous material, pseudostratified epithelium in the mucosa. Maybe you can guess that this is ciliated. We know we're in lung tissue because of the alveoli that we see. So that's going to be an easy leap to understand that we're looking at our respiratory type of epithelium and it's pseudostratified with cilia. Now the lung is going to be compartmentalized. Remember, our primary bronchi are going to come down, split to go to each lung. Secondary bronchi are going to branch, and then our tertiary bronchi go to a bronchopulmonary segment. Well, we can see in this particular slide how we have this lobule of our lung that is completely isolated with this connective tissue. So within the lung tissue itself, we are going to be segmented, and this is a lobule of the lung. We see many alveoli, but now we see these structures. At this magnification, I don't see any cartilage, so now we are talking about a bronchiole. And again, what level of the bronchiole we're at, we're not exactly sure, but we know it's not a bronchus because we don't see any cartilage. Now, higher magnification, we can absolutely see that we have our mucosal lining, and the cells are getting shorter and shorter, not as much pseudostratified now. Now we're more cuboidal to columnar. We can see our spiraling smooth muscle still on the outside. We don't have any cartilage. These are going to be our alveoli that are surrounding this. Here we have part of the arterial system within the lung itself. Now, we recognize our bronchioles because they have the spiraling smooth muscle, the respiratory mucosa, but they lack the cartilaginous plates. Now, we mostly just refer to them as bronchioles because primary, secondary, tertiary, and that hierarchy, we really don't know. We just know they are intrapulmonary. You won't find a bronchiole outside the lung, so even intrapulmonary is a little bit redundant. However, when we get close to the end of our conducting pathway, we can recognize, again, the structures of the bronchiole, spiraling smooth muscle, intact mucosa. But then we get to a point where we begin to see the interruptions of the mucosa as we lead into alveoli. Here we can clearly see one of those interruptions. So when we begin to see alveoli coming off of this pathway, then we know that this is the beginning of a respiratory bronchiole. If this then is the beginning of the respiratory bronchiole, then further upstream where we have intact mucosal lining is a terminal bronchiole. Now this one's kind of close because if that is actually a passageway leading to an alveolus, then we would have to also call this respiratory. But if we could look back even further and just see this continuous wall, this continuous uninterrupted mucosa, that would then be a terminal bronchiole that leads to and is adjacent to the respiratory bronchiole. And that's what we're looking at. Now, as we move from respiratory bronchiole, then we move into an alveolar duct. And remember, this is simply defined as a common elongated pathway that's defined by alveoli. And these alveoli that line the alveolar duct are going to have these rings of smooth muscle. And in cross-section, they just look like little knobs of smooth muscle. And so here we have an alveolar duct, and here we have an alveolar duct. And then all of the other tissue that we see here in this field are going to be actually alveoli. Again, another view of a bronchiole, we can see our spiraling smooth muscle. This looks to be relatively intact. However, if you look on the opposite side, you can see the interruptions to the mucosa. And with those interruptions, then we've got to say that this is going to be a respiratory bronchiole. We don't see the terminal far enough upstream. And as we leave that intact mucosa and we move into this area, again, where we see our smooth muscle knobs, now we're moving into an alveolar duct. A little bit closer magnification as we're getting toward the end. We still have our ciliated cells, and we can see them on the surface. You see they're not as tall, somewhat pseudo-stratified. We still have our basal cells, we still have our apical cells, and we can see our granules being released here, the mucus, but it's much shorter than what we're accustomed to seeing. Here we can see our smooth muscle running beneath. So this is still our respiratory type of epithelium. Here we have a little bit of a lower mag view of our intrapulmonary tissue, and we can see a nice elongated duct. Again, we're not seeing cartilage at this point, so we're going to call this bronchial. And you see how it tapers, the diameter tapers as you get toward the end. Here we have a branching, and again, you can see that the epithelium becomes interrupted at certain points until you finally lose it altogether. So we're going to say that this is terminal bronchial to the point where we may have a relatively short or absent respiratory bronchial moving into an alveolar duct. Here we have an alveolar duct. Here we have another terminal bronchial, possibly a short stretch of respiratory before we move into an alveolar duct. Now, from these points forward, we're just saying a bronchiole because these aren't extremely close to the point that respiration is actually going to begin. Now, I don't like them using this term, bronchus, because we don't see any cartilage present around this bronchus. So in this particular field, it might be on this side, but I'm not seeing any cartilage at all. So I don't know why they would put that there. Once again, a little bit closer look at that previous section. We have a lot of blood vessels present here. That's what we're looking at. But we have intact mucosa. So these are going to be bronchioles, no cartilage. Bronchial coming out, we begin to see the interruption. So we're going to say respiratory bronchial, which makes this a terminal bronchial. And then past this point, we have alveolar duct. Alveolar duct here. Alveolar duct here, with each one of these little compartments being an alveolus. Now, as we get closer to the end, as we begin to get in our respiratory area, we're going to see more and more of these dome-shaped, very tall cells lining this passageway. You can see we've pretty much lost the cilia. We're not pseudostratified any longer. It's almost a single layer of cells that we see. And these round domed cells look a lot like transitional epithelium. These are going to be our Clara cells that are on the surface of our airway. Little bit different stain. What we're looking at, the cartilaginous plates of two intrapulmonary bronchi. We can see our alveolar tissue here, so we're within the lung tissue. We have our mucosa, we have our bronchiolar glands in both sides, and then we have some blood vessels out here in the submucosa and in the adventitia. This is a really nice view of our very typical pseudostratified columnar ciliated epithelium in the respiratory tract. We can see the pink stain here. These are our mucinogen granules of our goblet cells. We can see our basal regenerative cells. We can see our tall ciliated cells on the surface. You can pretty much guess at this point we are looking at some point, some place in the respiratory system. Little bit lower magnification, looking at the inconsistent smooth muscle bands we see here in our submucosa. We have an artery here, muscular artery, possibly a lymphatic vessel present here, also some of our bronchiolar glands, and then our pseudostratified ciliated epithelium lining our airway. Now, as we get closer and closer to actual respiration, here we're beginning to look in our alveolar tissue. Now, many of the cells that we're looking at that line these alveoli are going to be quite flattened. So these are going to be our type 1 pneumocytes. And yet we're going to have these very tall cells that are going to be our type 2, our giant alveolar cells that are present, again, on the surface of our alveolus. And then we're going to have our resident macrophages that exist in the alveoli, and these are going to be our dust cells. As we look at more and more of our airway and alveoli, once again, type 1 pneumocytes, these flattened endothelial cells that line the surface. And then we see these larger cells that can be vesiculated. Again, they're going to be right on and part of the surface lining. And so these are going to be our giant alveolar cells, or our type 2 pneumocytes that are producing the surfactant. Once again, in this particular field, we have our type 1 pneumocytes. We have our vesiculated alveolar cells, type 2 pneumocytes. And then within the lumen, we have our much, much larger macrophages or dust cells. This is just a scanning electron micrograph showing our ciliated cells lining the lumen versus our mucus-producing cells. It's a nice view. We're very familiar with the structure of cilia already, the basal bodies there that we can see. We see cross sections in the axonemes through our cilia, and then in the very center, the darker mucus-producing cell. And lastly, this is a chart that a student in past semesters has put together, basically showing differences from our epithelium, the different cells that we have lining the epithelium, mucus-producing cells, glands, cartilage, elastic fibers, and smooth muscle. We're going to start in the trachea and make our way down to an alveolus, and as you can see, mucus cells tend to disappear, glands disappear, cartilage disappears by the time we get to our bronchioles. Elastic fibers are present but diminish drastically. Smooth muscle is gone by the time we really get to our alveolar ducts. Remember, it's just the knobs that we see at the beginning of our alveolar ducts. Epithelial cells, here's our typical ciliated that we see in the trachea. When we get to our bronchioles, we have the ciliated, but those dome-shaped Clara cells. Then as we get into our alveolar ducts, alveolus, sac, and alveolus, we have our type 1 and type 2. That should be the same there, type 2 pneumocytes.